Okay, so in this video what I want to deal with is the two pathways that aren't really about energy, getting energy, so much as they are about keeping a certain organ in our body happy, namely our brain. And those two pathways are gluconeogenesis and ketogenesis. Let's think about the brain. The brain is kind of an unusual organ in the body. First of all, its need for energy is very high. It weighs 8 pounds out of a typical 150-pound person, and yet it consumes 20% of the calories that you take in. It's a highly active organ. In spite of that, it is highly reliant on the rest of the body to provide it with energy. Part of the reason is there is this physiological barrier called the blood-brain barrier, which keeps everything out except for a few limited molecules. And in particular, fats cannot cross the blood-brain barrier. So that means that the brain cannot use fats that are available in the bloodstream, can't take it up, and they also can't use amino acids for fuel. They don't have the machinery to take those two potential energy sources and turn them into ATP. But what they can use is glucose and ketone bodies. That's it. So the way that the brain works is that it has its preferences. Its favorite food, its favorite source of energy is glucose. And when you eat a meal, within 2 hours, the levels of glucose that are present in the diet are enough. And that's actually why the glucose is in the blood, primarily is to feed the brain. But after 2 hours, after the meal, the blood glucose levels have cleared, and you are into the period of time where you're beginning to secrete glucagon. So once the dietary glucose is gone, then what has to happen is the glycogen that's stored in your liver has to kick in and start producing glucose, which then ships out to the brain. So as long as you eat every 3 hours or 12 hours or so, which is conveniently just about right for when you go to bed and wake up the next morning, you can keep a steady supply of glucose in your brain, in your bloodstream, which keeps the brain happy. Okay, but when those sources of glucose are not available, then your body has two alternative pathways for keeping your brain happy. One pathway is called gluconeogenesis, and the other is ketogenesis. So let's start by talking about gluconeogenesis. Remember that gluconeogenesis produces glucose from starting materials. This pathway is turned on by glucagon and it's inhibited by insulin. So it's a pathway that is designed to return blood glucose levels to normal under conditions when it drops. So what happens? Normally glucose is turned into pyruvate, acetyl-CoA, and then it goes around the citric acid cycle. But during gluconeogenesis, what happens is because there is no more glucose coming in, and remember there's no more glycogen coming in either because that's been depleted, then the body starts to use 3 different sources to make glucose. They are the glucogenic amino acids, here, anything that makes pyruvate or that makes any of the citric acid cycle intermediates. amino acids that make acetyl-CoA, the ketogenic amino acids, cannot be made into glucose. So those are some sources, some amino acids. Also remember that when you break down fats into triglycerides and glycerol, glycerol is that small 3-carbon sugar, and 2 of those can be strung together to make glucose, and that is considered part of the gluconeogenic pathway. The other place where you can get glucose from is oxaloacetate. So in the gluconeogenic pathway, all of these sources are turned into oxaloacetate, and then this enzyme here, which converts oxaloacetate into something else, so it's this enzyme and this enzyme that get turned on. And that's what makes it possible for you to turn around and take all of these intermediates and push them backwards towards glucose. Okay, I just want to point out here that this is one place where fats can be used to make glucose. But it's not actually the fats, like the fatty acids, that are doing it, it's the glycerol. And glycerol is not a great source of glucose because if you think about it, out of the how much energy there is in the fatty acids, you need 2 of those fatty acids, 2 of those fats to be triglycerides to be broken down into fatty acids to give you 1 molecule of glucose. Okay, ketogenesis. Where do ketone bodies come from? So ketogenesis is the process which generates ketone bodies. And ketone bodies are 3 chemicals here, which are collectively called ketone bodies, and they are very simple molecules. I'm not going to draw out their structure, we can just call it that. One thing that's kind of fun to know is that one of the ketone bodies is acetone, and acetone is the active ingredient in nail polish remover, and that's why when you are undergoing ketogenesis, someone who has a high level of ketone bodies in their blood, their breath smells like nail polish remover. So it comes from acetyl-CoA. And let's remember, where does acetyl-CoA come from? It can come from glucose. It can come from the ketogenic amino acids. And it can come from fats as well. So normally the acetyl-CoA goes into the citric acid cycle to make ATP, but if there's not enough oxaloacetate for the acetyl-CoA to bind to or to combine with, then the levels of acetyl-CoA build up. And under those circumstances, acetyl-CoA will be converted into ketone bodies. So we talked about what regulates gluconeogenesis, but what turns on ketogenesis? It is not regulated by hormones. This is one of these pathways that's just always ready and available depending on the availability of the substrates. And the 2 substrates are acetyl-CoA, really. And normally acetyl-CoA is pulled out of the cell because it joins together with oxaloacetate. That's a pretty hearty, vigorous reaction. There's a lot of drive for that reaction to occur. But if there's not enough oxaloacetic acid around for the levels of acetyl-CoA, then the extra acetyl-CoA is siphoned off to make the ketone bodies. So what turns on ketogenesis is a high acetyl-CoA to oxaloacetate ratio. So what causes a high acetyl-CoA to oxaloacetate ratio? Well, doesn't break down strictly under the fed and fasting state like the other pathways do that are regulated by insulin and glucagon, you can get ketone bodies formed during both the fed and the fasting state. So during the fed state, when it occurs, is when you are consuming a diet that has a high level of fats and a low level of carbs. Under that circumstance, your body is used to working with a blend of fats and carbs and proteins, and all of a sudden, all it's got to work with is fats and not much carbs. And so the machinery backs up with the level of acetyl-CoA that's present, and you will start to make ketone bodies. The other time when you get a high acetyl-CoA to oxaloacetate ratio is during the fasting state. There is a period of time when your body switches over from using its glycogen stores, and then it starts doing gluconeogenesis, but it needs to tap into that those stored fats. And when it starts to do that, beta oxidation pumps out a whole bunch of acetyl-CoA. And once again, your body is adapted to using glucose first. And so it takes a while for the oxaloacetate levels to build up to compensate for the high acetyl-CoA. So ketogenesis occurs during both the fed and the fasting state. So when is it that the oxaloacetate levels dip? How is it that those are depleted? If you think about, when you've depleted your glycogen stores, what are you going to do for glycogen? Your body has some interest in keeping those glycogen stores there. Your muscle needs them to be able to act, otherwise you'll be feeling pretty lethargic. So what your body will do is it will use that oxaloacetate to for gluconeogenesis, and then it will use the glucose to ship it out to the muscle, and it will be used to top off that glycogen again. Those are the two. All right, so if that's what turns it off, what is it that turns off ketogenesis? It's anything that boosts that oxaloacetate levels so that it matches what it is that the amount of acetyl-CoA that you're generating. And this takes time. How long? A couple of days. So if you ever shift from a diet that is a blend to one that is particularly low in carbohydrates or high in fats, it will take you a while for your body to build up its oxaloacetate levels. So where does the oxaloacetate come from? It comes from pyruvate. So pyruvate through gluconeogenesis makes oxaloacetate, and then that would go back up to glucose. But if you can keep that oxaloacetate instead of turning around and using it to make glucose, that will bring them back up again. So you can get them from amino acids, or if you get a little bit of carbohydrate in your diet again, then you can start refilling that pool from pyruvate. The other way that you can turn off ketogenesis is to reduce the load of acetyl-CoA. So once again, where does acetyl-CoA come from? Fatty acids. So if you get a boost of insulin, which remember insulin shuts off fatty acid degradation so that you can start to burn carbs again. So if this gets turned off, that will reduce the acetyl-CoA. If you get carbs in your diet so that this acetyl-CoA can be made into fats again, that will reduce it. And if you can switch to actually using your acetyl-CoA for energy production, which of course requires oxaloacetate. That will also reduce it. It's sort of circular logic.